So thanks everybody for coming. Today we have something completely different, but not super different. Thinking about how proteins fold and function. So Silvi has joined us all the way from the Whitehead Institute, very far away, and also at Harvard's microbiology department, where she's an assistant professor. Started at the Whitehead back in 2015 as a fellow, and in 2021 transitioned to assistant professor at Harvard Medical School. And her group has been really trying to study the structure and function of RNA in vivo. I'm not going to say any more about that because I'll let Silvi do that. We're really excited to have you. Thanks so much for coming. We do tend to interrupt during the talk, and I hope that's okay.
Yeah.
All right. Try to be
awesome.
very sort of engaging.
All right.
Thanks. So welcome.
Thank you. Thank you, everyone. All right, it's great to be here. And today I titled my talk RNA modeling, function, and engineering. And just to give you a brief overview, nope. All right, I'll use this. Nope.
Put it on the screen once.
Okay. Excellent. Just to give you a brief overview of what I'm going to talk about today, my lab is generally interested in predicting and modeling RNA structure, including alternative structures, and we're going to talk a lot about those today. And as well as RNA function, and specifically today we're going to talk about the RNA origami that determines mRNA translation. And the ultimate goal of the lab, one of the goals, is to build better RNA, but I'll save this for another time. So just like with proteins, RNA folding is hierarchical, meaning that we have a primary sequence that folds back on itself and into a secondary structure. And then with the help of yet additional interactions, we have tertiary structures. But unlike proteins, whose sequence, typically one sequence is deterministic of one particular 3D structure, for RNA, a given sequence, a single sequence can actually fold in drastically different secondary and tertiary structure. And this is just here a simple example, but they could be entirely different from each other, and they can have entire think of when we think about proteins. But most RNAs do have very stable secondary structures. And that tells us those structures tells us a portion of the RNA in 3D conformations it can assume.
So are you saying one secondary structure gives multiple tertiary, or you have multiple secondary?
So in this case, both are true. Both case, this secondary is deterministic of this tertiary, and this secondary is this tertiary. But this is a very special example because it has been crystallized, you know, and it's in PDB, and those are specialized RNA. In most cases, what you have is one sequence, multiple different secondary structures, and multiple different 3D structures that we haven't solved because they're way too dynamic.
Secondary will get multiple tertiary?
One secondary will give dynamic tertiary structures. Yes, the anticipation is that there'll be a lot of basically they will be different. Yes, how different? That's an unanswered questions, but yeah, you can go. So where's the field of RNA? And maybe that will help you a little bit in terms of protein structure. So I did a little research. And the protein structure field also started with secondary structure prediction. And there's a Chow-Fastman algorithm in 1975 that had, 55% prediction accuracy in terms of, alpha helices, the basic motifs, beta shaped stars. And you see for 20 years, not much happened. Finally, some big advances, of course, right now, the field of protein structure prediction is amazing. And we have, and this is just in 2020, now we have even more advances. In contrast, where is RNA structure prediction? So if we, here I'm showing you 4 widely used kind of best prediction softwares. And here's the F1 score saying how good they are. And the data set that we're using to ask how good they are is, can they predict RNAs that are deposited in PDB? So if we ask, can this software predict RNAs deposited in PDB? The answer is, oh yeah, pretty well. Like the closer you are to 1, that's the perfect structure. So actually, you know, if we're just interested in those RNAs, we are doing great. Unfortunately, if we ask, how well can this software predict the outside of PDB, the RNAs that we care about, and those are typically viral RNAs or messenger RNAs, so long non-coding RNAs. Now, all of this software suddenly precipitously drop in performance. And why is that? Well, one big reason is the data on RNA structure is really sparse and biased. So if you look at PDB, there are only 12,000 RNA structures. They're super short. They're, you know, less than 100 in length nucleotides. And also, if we look at other structures for RNA secondary structure databases, they're really highly biased towards very specialized non-coding RNAs, such as the ribosomal RNA and small RNAs like tRNAs and other small RNAs. And so what this means is that, you know, compared to protein structure, the RNA structure predictions are still tripping like it's the 1970s. This is where we are in terms of secondary structure predictions for RNA. Okay, so how do we go beyond that? I started working on this problem as a grad student, actually in Jonathan Weissman's lab, and developed a chemical-based probing of structure approach, and using this really small chemical dimethyl sulfate, because it enters in cells very quickly within a minute or 2, and it modifies the open bases, in this case, adenine and cytosine. So bases are Watson-Crick paired, they cannot react with DMS. And then what we did when I moved to the Whitehead is we developed a single molecule strategy where we modify a single RNA molecule with multiple open bases. And then we use a special reverse transcription enzyme. And when this reverse transcription enzyme goes through an RNA and there's a DMS methylation, it puts a mutation in the cDNA. And that's very convenient because all we have to do is then sequence the DNA molecules, and we know when there's a mutation relative to the reference, that is an open base in the RNA of that molecule. And next, what we did is to, so just to highlight, without the single molecule of probing, we can get really misleading results. And so imagine you have this RNA sequence that is forming those 2 very different structures. And it turns out this happens actually very often, we now know. But what happens is, you know, before this approaches, this could be happening in vitro or in cells, and you have multiple copies of these structures, of course. And here I highlighted the bases that are DMS reactive, so adenine and cytosines, and the bases that are this open in both structures, they're gray, because they're not going to be informative to us. But the bases that are open only in one structure or only in the other, they have their own colors. And so what happens is when you DMS modify any 2 random positions on different molecules, you end up with those reads that basically have this co-occurring mutations. But before our work, what was happening, what everyone was doing is, you know, they'll take all the reads all together and then, you know, okay, and then just count how frequently do you see this base mutated, that's your DMS signal, and then you're gonna call basically all those bases open because all of them will have DMS signal. But what we did is this algorithm based on expectation maximization clustering that instead of binning all the reads, it looks at the co-occurring mutations on a single read. So what it notices is that, oh, check this out. I actually never see this blue and yellow position occurring never occur together on the same read. They're always on a different read. And based on that, it's able to bin the reads differently and cluster them. And then we know, then we can get a very different DMS signal now, and we can generate accurate structure models. I'm not going to take you through all the validations. We validate this on Riboswitches and on known alternative structures. This work is published. Phil, the first author, is here in the audience. If you have questions for it, please ask him. But since then, you know, we've updated this. Now we have even better way of doing this. It's called seismic. There are other groups that also developed different clustering approaches. And the cool thing about this whole field is that these approaches have allowed us to uncover a lot of new biology. So in the case, in our first case, we worked on HIV and found interesting alternative structures that were regulating splicing. In the case of coronavirus, we found alternative structures that regulate frame shifting. More recently, in the case of mitochondria, we also found alternative structures that regulate frame shifting for mitochondria mRNAs. Another cool discovery was on human telomerase that has totally different alternative structures. Hopefully you can appreciate how different those are from each other. And that's an RNA that's been studied for over 30 years. And it's the first time we're actually figuring out it has a very different alternative life. The work I wanted to share with you today a little bit is unpublished on IRESs in human riboswitches. But before then, you know, we developed those methods that allowed us to find such cool biology. And so we asked ourselves, can we replace them with AI? Because, you know, AI is getting so good. And we'd like not to spend, you know, years and years pipetting and analyzing data. So well, we simply thought, okay, well, we're just going to generate a lot of data on RNA structure with our chemical probing method. And then we're going to learn from it. And we're going to learn to predict the structure based on this chemical probing data. And the key here, and actually a few groups did that at the same time, I should say. Riju Das did a very similar thing at Stanford as a part of a very big competition. But they focused on very short sequences, something like 140 nucleotides. And we wanted to focus on long sequences because well, it's not clear, but my hunch is that it's really important to look at long sequences, and they have different thermodynamics than the short ones. I can talk a lot more about this if you're interested. But the basic idea is let's collect all our data and then train a machine learning model to predict the 2D structure. So that's how we started making this database called rnandria.org. You can, you know, go on it. You'll see all the sequences, all the models that we've generated. They're composed of human primary microRNAs or just mRNA UTRs and things like that. And so you see a structure model, you know, their name, the chemical probing data, an accuracy number, 1 being perfect, and things like that. So in addition to this, we developed a new machine learning architecture. It was inspired by the Evoformer from AlphaFold 2 because that had just come out. And the new thing about this architecture is really just adding this self-attention layers. So people had already been using convolution neural networks to predict the 2D structure. It's the self-attention that was new. And we thought by adding self-attention, perhaps this network can better handle long distance interactions and can better predict structure for long RNAs. Yes.
Sorry, the question about the last step. You were calling 2,000 nucleotides short.
No, 2,000, I'm calling them long. That's an excellent question. And like, ideally, we want to probe the structure of the full length molecule as it exists in cells. And for mRNA, the average is like 1 kb, right? For some viral RNAs, for the coronavirus, it was 30 kb. So we don't know at this point, like how long range the interactions can be. I didn't go into it, but in the coronavirus, we found interactions as far as 1.2 kb apart. So they could be very far apart, and all the known algorithms have been optimized for local interactions. Yeah.
We had to stop.
Yeah,
So, AlphaFold with, or this analogy with AlphaFold.
Yeah.
And your slide about covariation.
yeah,
Sort of makes me think, I mean, the reason why AlphaFold is so good is because it has access
covariation.
to these
Yeah.
protein sequences, and you can extract the covariation. I don't really know, obviously those sequences come from DNA databases.
right.
I don't really know how big the RNA databases are, but if they, is that another approach? If you
Yeah,
think of covariation from RNA?
it's a great question, and people have tried. It turns out, for reasons that hopefully will become more clear to you as I go on, it's really difficult. One is because of obviously the coding region, it's the signal is overwhelming by the codons, by the amino acids. The proteins are beating us here. So there's any covariation, it's much stronger for the protein conservation. But it turns out in the UTRs can also be very problematic. And that is because, as I'll show you in the case of viruses, it turns out you can actually have the same function, but a very different structure for it. And we're still trying to understand, how that evolved, but it's been, no one has successfully been able to, and people have tried this, Elena Rivas, Sean Eddy, and they've done a lot of work on this. It works for super conserved RNAs like the ribosomal one, but we already have those in PDB. It works for the exact same ones that can also be crystallized, basically, or cryo-EM. Okay, so just a very quick, how do we train this model? We try to get obviously as much as data as possible. So part of it was just sequences from RNA Central, the Rijudas Ribonanza competition that had chemical probing data. And then we fine-tuned on our little data set because we had long RNAs, but they were much smaller in number. The testing is an important component here. Of course, PDB is a gold standard test, but as I said, it's not very useful in practice. So we very carefully curated structures from the literature that had been determined through various methods, and we try to find structures that really two labs that agreed upon and had done some kind of mutagenesis on to say, okay, well, we'll take this as a gold standard, because actually the biggest problem of the field is having this gold standard structures, knowing the ground truths, which is very difficult for RNA. So you see those are small number, but very important. So what we found in the eFold performance, basically, again, here I'm showing you the same thing that I showed you before, but now I'm adding some new players like Spot RNA and uFold. Those are purely machine learning based approaches for structure prediction. But you see the trend is similar, where they do really well on PDB, and when you challenge them on the things you really care about, the performance is very poor. And so with eFold, we're excited to see a pretty significant increase in that performance. It's not where we want it to be yet, but at least we're in the right direction. And so part of this, we were asking, okay, is this performance because we added some new data in the training that those guys didn't have, or is it because of the new architecture, as I said, the self-potential layers? So it turns out it's both, in short. And the way to test this, for example, to test the architecture specifically, we took 120,000 sequences from 6 families. And then we trained uFold, which just has convolutional layers and networks, and eFold that has a self-attention. So 2 different architectures, but trained on the exact same data and asking how well can they generalize. And what we found is really eFold architecture performs better only on the 16S ribosomal RNA. And that happens to be 5 to 10 times longer than any of those other RNAs. And it has many more longer distance interactions. So we do think that based on this, the self-attention is helping with long distance interactions and for long RNAs. Training data increases. Of course, the more data, the better the performance. Here is just showing how as we add more data, the performance increases. And so this is kind of giving us hope that if we have more money and more data, one day we'd be able to, you know, bring it up to some reasonable thing, because I'm not gonna tell you to stop what you're doing right now and go do RNA structure prediction with eFold. It's not quite there yet. Yeah.
So why is the PDB RNA seemingly easier? I assume you're not training them, so. So is there something about the RNA and the PDB that just makes all the programs predictive level but not the other kinds of RNA?
They're very short. So, I mean,
Very short.
there are not that many possibilities. As you see, most of them are like 80 nucleotides, or they're under 100. And some of them are like microRNAs, they're 20. So the possibility of who's pairing with who, and here we're just looking at secondary structure, right? So actually, there are very few constraints when they're so short. So I really think it's mostly the length.
We also.
Yeah.
We also that it's just super stable because people.
That is very, yes. Yes, great point. Yes, also because, yes, they're super, super stable, so everyone can predict them.
And what do you mean
yeah.
by structure? You said that there's many different secondary structures. So what is it? Like you said it's 10%.
You're paying very close attention. So for this, basically, the ones that are in PDB, they're mostly one structure because they're so short. The other ones, I hid this because I didn't want to get things overcomplicated. But actually, we specifically looked at regions that just form one structure. So we took everything out that we see is forming multiple structures. And we only took the ones that we, you know, are pretty certain, we can't be 100% certain, but we're pretty certain are consistent with one structure. But you're absolutely right. What we really want to be able to do, I showed you all the great biology we discovered was because of the multiple structures. We want to be able to predict multiple structures. That's further away just because it's even less ground truth available there.
Multiplication methods.
But we are trying that. Yes. Yes. Yeah.
It says, how do you split the data set for training and testing?
How did we split the data set? Let's go back. Oh. I mean, basically, in different ways, depending on what we were testing and what, but basically we did look for sequence similarity, I should say. I mean, there is one problem that plagues the field as well, is that you look at sequence similarity and you throw away to make sure that nothing is, but you use a cutoff, is that nothing below 20% similarity, you know, I'll throw it away. But then it turns out that, I mean, you can see that a lot of them bleed in because the structure is the same. The sequence is very low similarity, but the structure is the same. So that's another problem that all of those approaches have.
Here you split by sequence similarity, like either cutoff.
We filtered. So all those that we've used for training and fine tuning, we specifically have filtered, and we had to filter it actually twice the second time after review, because reviewers were like, oh, we don't think you filtered enough because no one filters enough. And then we're like, oh, look back. So we had to filter twice to get rid of as many as possible. But actually, if I don't know if I have this slide, I just want to show you, but you're absolutely right. Okay, maybe I do. So if you look at uFold, look how well it predicts on PDB. And then when we retrained it, it actually, no, I don't have the slide. But basically, when we took the same architecture and we found out that a lot of algorithms have been contaminated with their training sets, having structures from PDB accidentally. We'll fizz around anything that's 20% similar by sequence. Yeah.
Like, oh, kind of look at this P&L, which basically looks at how many of the real contacts are recovered, the ones you take the top out contacts. So how is the prediction of like that?
So that's kind of built into the F1 score. Like we score how many correct base pairs are you predicting and how many wrong ones. Sorry, I should have explained that score better. Yeah. But you're right, there's space for improvement here. There's space, we have tried to do that too, to decompose the F1 score and see, okay, can we do better? And yeah.
Like a mass token kind of prediction, or you're just predicting your secondary structure at all? Let's see.
We are predicting the secondary structure in this particular case.
Well, or just if you go to your table of different data
Yeah.
sets, I was wondering, like, the first row there.
Yeah.
Like, what is that, what is the, or?
Yes,
what are the-
sorry, you guys are getting into a lot of details that I skipped because I want to get into, so, okay, so basically what we decided is, look, we know that thermodynamic, so I'm gonna give you the full answer, so hang in there. We know that thermodynamic based approaches like RNA structure, that they have empirical measurements, thermodynamics. They are actually the gold standard right now for secondary structure. They're the best. Even if you, so any of the machine learning ones have not been as good. So we're like, okay, let's learn, in order to improve upon the best, let's first learn to be as good as RNA structure and then try to beat it. So the way we're pre-training here was simply saying, okay, let's take those sequences from RNA Central. They were just sequences of viral RNAs, human RNAs, just any RNA sequence, and then ask RNA structure to predict them. And then we're taking that as ground truth prediction. We know it's not, but it's the best one out there. And we're like, okay, if we train it on this and then we can fine tune it on something better than RNA structure, maybe we can beat RNA structure. That was, yeah, I did not explain that logic whatsoever, but you guys.
This whole sequence, the label that makes this structure.
Yes, yes, correct, yes. I don't know how that compares to what you guys do because I'm not sure exactly what you do. But those questions are super, super good. I've never gotten such detailed questions. All right, so we need more data. As I said, the biggest problem is we don't have ground truth. And ultimately, RNA structure is confirmed by function. That's the easiest way, is to tie it to some function. You know it's functional, then you think, okay, the structure must be the right one. And so that's why we started to collect more data specifically on functional RNAs. And what I'm going to tell you about is RNAs that are really important for translation, including IRESs and human riboswitches. So mechanisms of translation, very quickly, for all human mRNA translation, typically it's cap dependent. What that means is that a bunch of factors bound to the cap, and that recruits the scanning ribosome, and then you get translation. In contrast, IRESs are internal ribosomal entry sites, so you don't have any cap, and you just can directly recruit either the ribosome or initiation factors and the ribosome, and that's how you translate your mRNA or your genome. And viruses use this as a really nifty strategy to hijack the host ribosomes because they mess with cap binding proteins, and then they can use all the ribosomes just for themselves. Makes a lot of sense. So we specifically focus on piconaviruses because it's the biggest family that has irises, and this is the whole genome. It's only 10 kb, and it's all one open reading frame. It's kind of cool. And one AUG, and it's absolutely controlled by this one iris. And just some examples: they infect human, they infect animals. I'm going to tell you a little bit more about them later. But basically the way they work is they get in the cell and then they literally inject their positive strand RNA genome. And with this iris, they use it to recruit initiation factors in the ribosome. They translate their whole genome into one big polyprotein. They cut it up, and then they make their virions, and they go out to infect other cells. But what if what we're realizing might be happening quite a bit is that the virus gets in, but then actually maybe this iris misfolds, or for some reason it's unable to recruit, the ribosome and initiation factors. So in this case, the iris becomes a gatekeeper for productive infection. Right, so the function is really important. And what we want to know ultimately is what RNA structures determine this iris activity. And we want to use those because, as I will tell you about, there are 200,000 different irises. But 200,000, and machine learning people get excited about this number. Okay, maybe we can learn something from there. All right, so what is known in the field? In the field, currently there are 5 different IRES types, and you will find this very funny, I think, because none of them, for piconoviruses have been, there's 3D structure. And actually, they've been classified by secondary structure motifs, is the way they've been classified. Oh, well, this one has a cruciform, like right here. So we'll call this one type 1. So the irises really are in the 2D state. The big problem has been because they're long. So here we're talking about all the piconoviruses are longer than 400 nucleotides. So they're between 400 and like 1.5 kb on the longer side. So that's why there's no full length. Little pieces have been crystallized and cryoed. But just to tell you, that's how they're classified. That's when they were discovered as types. You can see type 5 was discovered as recently as 2011. And the classification is, I would call it a little bit loose because it's really based on, is this motif present? How many domains do we have? Like, things like that. And a lot of structure predictions. But an interesting thing is that, if we take just the coding sequence, so this is of 96 species, we cluster it by coding sequence, what you see is that actually the IRES type doesn't cluster together as the coding sequence. So even though those are very similar viruses, they have different IRES types. And white color is undefined IRES type, it's just unknown. This to say that, there's evidence, the literature thinks that the 5' UTR of piconoviruses can frequently recombine. So what we were trying to understand the modality, evolution, constraints. Yeah.
Yeah, so
Yeah.
what do the irises bind to? You have this picture of this blob.
Yes.
Brown blob. What is that?
Brown blob. Oh, yes, sorry, this one. So depending, so what exactly. So great question. So the irises can bind a subset of initiation factors like EIF2A or 4A, like a small subset. And depending on the iris, it requires either 1, 2, or none. Like the most dramatic irises require no, they directly bind the ribosome, but those tend to be the ones that actually don't work so well. They give you just a little bit of translation. The ones that are the best working most likely bind multiple initiation factors, and including some of them bind what's called ITAFs.
Can't you classify according to what they bind to?
Yes, great.
There's hundreds of thousands of them.
Absolutely. And yeah, a lot of them are still unknown what they bound to. Actually, most of them are still unknown. But you're absolutely right. The way people have been classifying the irises is they typically start with a structure prediction, and then they try to see, okay, is it this factor dependent? To get, so they've been classified one by one. There's no systematic approach to classify them, and paper by paper. But yes, absolutely. And we still don't know all the factors that they bound to. And that's part of what we're going to try to do. But today I focused it only on RNA. So that's why I didn't explain anything about the brown factor. All right. So as I said, a very small number have been functionally characterized. Here I'm showing hepatitis A virus. Multiple groups have shown that, for example, it has very little activity in HeLa cells, but in liver-like cells, it has much higher activity, which makes sense and could be contributing to the tropism of hepatitis A iris. But again, people have been looking at those irises one by one. So it's really slow. And so we're like, okay, how can we do this in a higher throughput? Now, the problem being is that they're really long and that we don't want any cap-dependent translation to interfere or read through, ribosomal read through. So in order to do this, we actually did it in circular RNAs. So we designed a library, so it's a circle. So the only way to get translation is to get from the inside. And the insert, again, is, the whole iris, which is pretty long. And then for our pilot, we took 96 from viruses that have different hosts. So half of them were human, but the other half were infecting, toad, seal, chicken, goat, etc. And then we asked, okay, let's look at those. Let's figure out what their RNA structure is, because, it's unknown, it's only been predicted. And then let's measure their translation efficiency by polysome profiling across different cell types. And this is the work of great postdoc Adam Sikla. So what we immediately saw is this widespread of cell type specificity. And here I'm just showing you translation data. Well, all you care about here is when you see red, that means it's highly translated, and blue, it's not highly translated. And this is the different iris sequence. And so, for example, this cluster of 3 irises here is very highly specific and translated in human neurons like cells, but not really in other ones, other cell types that we tested. Alternatively, there was one that was very specific for monkey kidney cells and not the other ones we tested, and so forth. So there's a lot more cell type specificity that was previously anticipated. We don't know what it's coming from. It's probably a combination of RNA structure and protein binding factors. Yeah.
Is it known what happens to viruses when they are circled?
Yes. Yeah, great question.
Big issues, like I think they call it cargo. If there's like cross-talking with the iris and the cargo.
Great question. So the only thing I can comment on for now, well, I can comment on a couple of things, is that we probed the structure of the iris in the linear versus the circular form, and it was exactly the same. I emitted data, it looked the same. So that we were very happy about that. The activity reported typically correlates, and we've done those experiments as well. There's a company, Orna, that we briefly talked to. They do a lot more of this in the high scale. There's also Alex Wesselhoff. He does the circRNAs for us. Actually, he used to be at MIT. So basically, they also see similar thing. What I want to say is, for the most part, they correlate in the linear and circular form. They are outliers. They are exceptions, but for the most part. Okay. So here's the other very fun thing that we found that, company already knew for sure, but will never publish most likely unless they're watching. All right, but yes, so the one most commonly used IRES actually in bioengineering is EMCV. And actually, it's a pretty weak iris out of is just even 96. And so this is data from HEC293 cells very specifically. And now we can look at this iris activity by iris type. And what you immediately see is, okay, if you're doing bioengineering and you want high translation, don't use EMCV because that one's pretty low. But actually, this super cool result that this very newly discovered type 5 irises is really predominating our highly translated ones. And type 5, we know the least about. We actually don't know all the protein factors required for type 5. There may be a couple known or so. This is just to answer the question of why, one question of why we can't use, things like covariation, because really, I'm not expecting you to get much from this. This is if we make structural models for the 96. I mean, all those 96 irises give you translation, and their structures are entirely different. Like there's very little things that they have in common, and we're still trying to figure out, what are the things that matter. And obviously, some of them have been previously characterized, so we're going off of those.
One of these maybe has 10 different confirmations that have different things.
Yeah. So we're looking into this, and actually we haven't done the analysis for this particular irises, but I collaborated with Michael Marr at Brandeis, looking at a Drosophila iris, and it was actually astonishing. They did not have any alternative conformations, neither in vitro nor in cells. They were super stable. They were exactly the same. So they were much like a ribosomal RNA, actually, but probably haven't been crystallized just because they're too big. And, with the ribosome now, you can resolve it because, it's the whole ribosome and it's with the proteins. So those, people who are doing cryo-EM, I think they should give it a shot for some of those irises, because especially with proteins that can, make it, yeah.
From this picture here, if you were to analyze minimal motifs that might be essential for initiating translation, that might be common. Do you see anything? Is there anything that jumps out, or it's just, there's nothing in common in all of these secondary structures?
No, there are things in common, and Adam is doing a lot of, he's actually using like graph theory to simplify the motifs and then see what's it's just more complex than we would like it to be. But yeah, yeah, there's stuff in common. We're trying to now yeah, we're trying to maybe that might be too few to get at, unfortunately, the 96 also. So it'd be nice to scale up some of them. But this is kind of the motivation behind some of these motivations is this, the coronaviruses or these viruses that have a very high pandemic potential, but very low existing countermeasures. And for those of you that don't do viromics every day, it's been a giant kind of boom in viromics because now people very constantly are sequencing all kinds of tissues from all kinds of animals, and we have so many sequences deposited, and that's why I wanted to say for the coronaviruses, we have 200,000 unique genomes in there. And then we know that they recombine a lot, but we really don't know anything about, the function of them just by having the sequence. And so kind of our goal in this is really to start exactly what you said. What are the essential RNA structures in this? Can we therapeutically target them? Can we, use this when we make good models at this scale? Can we use this for machine learning? And ultimately, can we get something more by sequencing a virus from a chicken? Can we say, oh, wow, this virus is actually going to work really well in human cells? Because I didn't tell you this, but those viruses here, they're the best working in HEC203 cells. They come from a goat. They don't come from human. There's, I think, one that comes from human, but the top like 15 are all from goat. Yeah.
I have a question. Sustained by work for that particular thing.
Ah.
Are you getting functional activity as you seeing signal go up, or are you seeing anti? Because a lot of them are RNA.
Oh, you see what? Anti what?
Like anti-expression right there, because a lot of these RNAs are actually silencing. They're actually doing inverse. They might actually do a lot of regulation.
They're silencing. So the way we're asking them, we just have a reporter. Well, we're asking two different ways. One is just polysome profiling. So we just see them loaded with ribosomes. And that's what I'm saying works. It's loaded with ribosomes, but we also confirm it in linear form or even with RNA transfections in multiple different ways. Like our top hits, we've confirmed in multiple different ways. We're just transfecting RNA. Yeah. So it's making protein. We can see it making protein.
RNA also has a lot with silencing due to lysosomal.
Silencing because of what?
Because you're interferes with ribosome effect.
Oh, it interferes with ribosome like scanning or?
Like actually ribosome, so it doesn't go through the culture using things. If you're a nice study, they're working a lot of that when you actually show that some of these campers science.
Ah, some individual. So the ones that we're taking are from viruses that are known to, infect those tissues. So they're mostly, those are mostly just trying to recruit the ribosome and initiation factors.
The same long RNA. Probably all the foreign structure stuff right there, but it's constantly confounding space right there.
Oh, with evolution, yeah, yes. I mean, it's been really hard for sure, to look at this with evolutionary constraints, unfortunately. But it's something we are thinking about, definitely. All right, so I think I still have a few minutes. And this is great because I'm just gonna give you a tease on the human riboswitches. So now you know everything there is to know about irises, hopefully. And the other data set that we want to, we're really interested in is this RNA riboswitches. So what are riboswitches? They're sequences that change structure upon binding a ligand to control gene expression. So, you have one structure, unbound, you bind something, it's a small metabolite, and then boom, no gene expression. This could be either on transcription or translation. And this is kind of, this metabolite can be so many different things. It's pretty spectacular. It could be as simple as an elemental ion. It could be, RNA derivative. It could be a sugar. It can be, a signaling molecule. But all those beautiful molecular switches that really can turn genes on and off so nicely, the presence of small molecule, have only been found in bacteria thus far. And so we and others, I don't know if you guys have heard the talk of Jared Brumder, have been searching them in humans. So how does this work? Basically, what we did is we took a conserved set of human UTRs, in vitro transcribed them, the nature refolds, and then we do DMS map seq without addition of a ligand. And then we add some small metabolite and do DMS map seq again, and then simply ask, well, did anyone care change structure because we added this thing? Very simple question. And this metabolite, we tried a lot of different versions of this. So very quickly to just show you, this works really well for known riboswitches, like the adenine one. So if you look at DMS reactivity, in the presence of adenine versus the absence, and this is a single nucleotide, but what you can see is, it changes. That's all I want you to see. The DMS reactivity changes. And really does correspond to drastically different structures now. You see this one is kind of this, and this one is 2 guys, and this one is 1. When we have replicates, obviously data is really nice. So then when we have library of thousands of things, all we look at is this R-squared number that tells us, okay, the structure stayed the same or the structure changed. Okay. And so I'm going to show you one of our screens with, so this is the DMS correlation R-squared, and this is the sequence rank. So this is about, 2,000 sequences. So here each dot is a sequence, and what you can see when you add amino acids, actually the vast majority of sequences don't care at all that you added amino acids in terms of their structure. It stayed the same. The DMS correlation was very high, but some of them really cared. They changed very drastically. And we ask for specificity such that, if you change structure, you can be promiscuous. If you change structure with amino acids, I already told you, then when we add nucleobases, then you're not going to care about it. You're only going to very specifically care that we added amino acids. So this is the kind of screening that we did quite a bit of. We have dozens of hits that are interesting. And then what happened is, yay, all our friends got terminated. So at this point, we had to pause on the vast majority, except we follow up on one. And I wanted to kind of tell you the story, the end, well, not the end, the middle of the story for one. The reason why we followed up, because it was a 5' TR of a cancer-causing gene. And what our data suggests, and I think this is very cool, which is why I wanted to share it with you, is that for this gene, actually, when you have the full 5' UTR, under low concentration, nothing happens, and this message is translated, and you make this protein. But when you have high concentration, actually what happens is this mRNA comes together and dimerizes. And that prevents now the ribosome, and translation is shut off. So it's kind of like a break that you can never make too much of this protein because of this built-in break. And what we found is that, if you make this dimerization mutant, or because it's a cancer-causing gene, presumably in perturbed metabolic environments, now even at high concentration, you don't dimerize anymore, and you make a lot more of this protein, and that's a protein that helps you metastasize and spread and so forth. So we're excited about this. It's totally new mechanism in terms of translation regulation in mammalian cells. There are a few other genes that suggest similar strategies. We'd love for this to be, more than one, and maybe more widespread. as a way to regulate your protein output. And of course, it opens up therapeutic applications. So as kind of the summary, we are interested in both irises and riboswitches as a way to engineer desired proteins in therapeutics. And irises are particularly interesting because of their tissue specificity of expression, and riboswitches because you can get very precise on and off in the ideal case scenario with a small ligand. So we went through all of it. I really want to thank you guys for your attention. No more energy on me. But you know, a lot of people in the lab actually, and a lot of our collaborators, and funding is essential. The funding we have remains. So yeah, thanks for listening. Tammy. Yeah, it was all done in vitro because it's so hard to control the concentration of ligands in cells. And yes. We might, yeah. Yes, yes, yes. But yeah, there are a lot more that can be done there, definitely. Yeah.
Super exciting.
Thank you.
I think the tissue selectivity is also just really fascinating. I'm sure there's so many variables that are leading into that, but I would love if you could comment maybe just a little bit further. What are some of the factors you think that are driving that selectivity?
Well, right now, as far as evidence in the literature, is that those are IRES activating trans factors, this called ITAF. So there are just some proteins that specifically are expressed more in one tissue, or a lot more in one tissue versus another. And how do they do this? The best, I mean, I'm just telling you based on the field, the best argument is they're actually helping the IRES fold in that particular tissue. But it's pretty open question, and there are very limited number of experiments that have done to address it. As I said, because you know they've only looked at one iris at a time and one. Yeah. Okay, I don't know. Sorry.
So I had a question about the dynamics of these, like either riboswitches or the other confirmation structures. Obviously, they have to be stable enough so that when you do the DMS, that you're just capturing one form. But you can imagine that at some extreme, they're going to be interconverting so fast you won't be able to detect it. Has there been any work done to try to see how you can use those either dynamic properties to somehow explain a little bit more about the structures themselves?
So it turns out that most of the structures that we look at in terms of the secondary structures, they're actually very large transitions. You have to break multiple base pairs. So every single base pair could be like 1 or 2 kilocalories. And so because you have to, in order to transition from one to the other in vitro, you have to break 6 base pairs, for example, and then reform. And so it seems that's very unlikely, energetically unlikely to happen in vitro. And so that's why, and you're absolutely right, that's why we were able to capture them. In cells, what we often see something that looks like denatured, that's actually modified pretty uniformly everywhere. And I think there is definitely a possibility that that could just be an RNA that we're in the process of converting from one to another. And then, you know, it got binned all together with this state. But basically the reason why we can capture them, because you actually need quite a bit of energy, they're pretty stable. And they're gonna, yeah.
And so you run those at lower temperature too?
We do. There are effects depending on the structure. There could be small effects or very big ones. So yeah. Oh, okay. How about in the back? Okay.
Thanks so much for the great talk.
Yeah.
I was wondering if the beautiful IRES structures that you showed, is it possible to sort of probe structure activity relationship with respect to those secondary structures and like translational activity and then sort of locally returning structural elements to like areas or something, or is that tricky because like even a local disturbance can kind of like result in like a completely different thing?
No, I think you're absolutely right. That's what we do. That's what we would do, you know, if we have money. We would do some local perturbations for intelligent designs. And actually, someone in the lab is already doing that on a small scale to understand exactly what you said. And from our experience, a lot of local perturbation remain local. But it's sometimes the local perturbation is global, but that's more rare. And it's also very interesting because it tells you about the long distance. But yeah, you're absolutely right. That's
Riboswitches primarily bind small inner compounds, or can they also bind higher
Oh, great. Well, they do have some chiral specificity, just if you look at this table, as far as I remember in bacteria. So you have to realize this is all from what we know from bacteria, because that's where they're the best. Oh, I went too far. Characterized. They're quite specific. I'm quite sure that some of them have chiral specificity, but I don't know. You know, of course, some of them are just ions. So, oh, for our human ones. We haven't looked. We haven't looked. But because there's, yeah, we haven't looked. That would be very interesting. Yeah. Yeah.
Thank you for the great talk. When you talk about the prediction accuracy of people,
Yeah.
do you know whether when it, for example, gets it wrong, does it predict a pretty reasonable structure that could definitely be one of the other like possible configurations of the RNA it's into, or is it pretty much completely random? Is there a way of telling that?
Yeah, that's a great question. We've been trying to actually, one way we're trying to improve eFold is by trying to figure out what data do we need the most, actually. So where does it get it the most wrong? And can we collect that data? You know, or even actually what would be great is even if it could be like AlphaFold, give it some score. I'm really not so certain about this one, you know, just but the short answer is it's a mixed bag, and we don't have enough numbers to tell you, you know, significantly it's one way or this way, like because the numbers of ground truths are so small. So I can tell you 10 go this way and 5 go that way, but what does that mean? So great question though, and something we're definitely trying to understand better. Yeah.
Really good slide out. So my question is about metabolites. So in cancers, it's kind of well known that cells can have their metabolism hijacked.
Yeah.
Right? And there's oncometabolites, like 2-hydroxyglutarate. And I'm just curious, has anybody looked at these oncometabolites, and can they bind to RNA switches and start to activate or inactivate expression?
Is someone making them in high enough amounts? Those metabolites are
They can be millimolar.
Yeah. I mean, basically the ones we looked at are the ones we could purchase or the ones we could get from our friends.
Oh, you're talking about, is there some?
Yeah, exactly. Yeah, very practical question here. But those will be.
Cells make them in high.
Yeah, we are looking at our top candidates. We are looking at the structure in different cell types, including like comparing normal and then the same cell but cancerous. So trying to get indirectly at that. But the ones we've tested are just the ones we can just get our hands on. And if you know other ones, let us know. Yeah. That would be awesome. I think that would be.
My second question was, is John Jumper in your lab?
No, but he was very kind. Yeah, he took this picture with us and wore the shirt. Which was that's John Jumper right here. You recognize him. He looks like a lab member. I told him he can join. He said, Sylvia, I'm sorry, but I already have a job that I kind of like, and I have a lot more compute than you do. What? was, so no, unfortunately, no, was from the Express.
More questions? Well, thank you. Sylvie, again, it's your great.